Every living system runs on a thermodynamic deficit. Building complex organic mass out of a low-energy inorganic gas like carbon dioxide requires a massive, sustained input of energy. In plants, this conversion forms the chemical foundation of the global food chain. These four charts establish the strict environmental limits of this planetary engine. By plotting the rate of photosynthesis against external conditions, we establish four kinetic curves: light intensity, carbon dioxide concentration, temperature, and water availability. Observe the mathematical signature of these graphs. Light and carbon dioxide concentration hit plateauing asymptotes. Temperature shows a sharp drop-off past its peak. Water creates a rigid threshold crash, where the rate collapses when supply dwindles. These macroscopic environmental limits are dictated by specific microscopic biochemical bottlenecks operating inside the chloroplast stroma. This pathway was mapped in the 1950s using radioactive carbon-14 isotopes. Researchers, including Melvin Calvin, proved carbon fixation is a concrete structural reality. They identified a three-phase thermodynamic engine known as the Calvin-Benson-Basham cycle. The process is divided into carboxylation, where carbon is captured, reduction, where energy is injected, and regeneration, where the starting molecules are rebuilt. The cycle cannot convert multiple carbon dioxide molecules into sugar in a single reaction. It is a strictly stepwise process reliant on the turnover rate of specific enzymes. To understand global plant productivity, we must systematically deconstruct the cycle. Tracing each of the four macroscopic kinetic curves back to their microscopic phase reveals exactly what limits the speed of carbon fixation on Earth. This chart plots photosynthesis relative to carbon dioxide concentration. The curve rises linearly before hitting a definitive plateau. This limit exists in Phase 1, carboxylation. Here, the primary enzyme, rubisco, catalyzes the binding of a 5-carbon molecule, RuBP, with a single molecule of carbon dioxide. This creates an unstable 6-carbon intermediate called CKABP, which instantly cleaves into 2 molecules of the 3-carbon compound 3-PGA. This chemical reaction is constrained by physical space. Rubisco has a finite number of active sites. As carbon dioxide concentration increases, these sites progressively fill. Eventually, every available rubisco enzyme is engaged in a reaction. When the enzyme reaches this maximum catalytic turnover, additional ambient carbon dioxide cannot be processed because every active site is already occupied. Plants attempt to bypass this physical bottleneck by increasing the sheer density of rubisco in the leaf, making it the most abundant protein on Earth. The macroscopic carbon dioxide asymptote is the direct manifestation of rubisco reaching its maximum catalytic capacity. This graph compares light intensity to the rate of photosynthesis. Like carbon dioxide, it shows an initial climb followed by a hard plateau. The bottleneck here is stoichiometric. During Phase 2, reduction, and Phase 3, regeneration, the cycle consumes energy currency. Fixing 3 carbon dioxide molecules to yield 1 net G3P molecule requires exactly 9 ATP and 6 NADPH. While the chemistry of the cycle is technically light-independent, it relies entirely on the ATP and NADPH produced by the thylakoid reactions. Referring to this process as the dark reaction is a structural misnomer in biology. The cycle is governed by the thioredoxin-ferredoxin system. When light hits photosystem 1, it reduces ferredoxin, which then reduces the thioredoxin protein. This activated thioredoxin severs a cysteine bond found on key Calvin cycle enzymes, toggling their active sites to an operational state. This toggle ensures the cycle runs only when light is available. Running in the dark would wastefully respire intermediate molecules and drain the plant's energy reserves. Light dictates the Calvin cycle by providing the raw stoichiometric energy and by acting as the structural master switch that prevents catabolic collapse. The temperature curve begins with a predictable upward slope. Higher temperatures increase the velocity and frequency of collisions between rubisco and its substrates, accelerating the reaction rate according to standard chemical collision theory. However, past the optimal temperature point, the curve shows a sharp crash. Extreme heat eventually causes proteins to denature, but the primary culprit for this collapse is a limitation in rubisco substrate specificity. Because oxygen and carbon dioxide are structurally similar, rising temperatures cause rubisco to mistakenly bind oxygen. This process, known as photorespiration, turns rubisco into an oxygenase. Binding oxygen yields 2-phosphoglycolate instead of the 3-carbon molecule needed for the cycle. Salvaging this molecule requires the plant to expend energy, ultimately leading to a net loss of previously fixed carbon. This catastrophic temperature crash is the macroscopic result of rubisco's deteriorating specificity. Heat turns a mass-building synthetic engine into a mass-wasting catabolic drain. When we evaluate water availability, we see the impact of environmental water deficit. The plant triggers an immediate, aggressive mechanical defense against severe water loss. The leaf's microscopic stomata violently swell completely shut. Stomatoclosure halts the intake of atmospheric carbon dioxide. Internal carbon dioxide concentrations plummet, starving rubisco of its primary substrate and arresting the carboxylation phase. Extreme waterlogging causes a different failure. It reduces soil oxygen, halting root respiration and crashing the cycle. Water availability limits the rate of photosynthesis by physically obstructing the physiological supply chain of carbon dioxide, bypassing the Calvin cycle's chemical equations entirely. 3 carbon dioxide molecules in, 9 ATP and 6 NADPH in, 1 net G3P out, 3 RUBP molecules regenerated. The cycle is bound by active site availability, redox toggles, kinetic specificity, and substrate delivery networks. Mapping these precise kinetic limitations provides the baseline framework for modern agricultural bioengineering. Historical agriculture focused on brute force scale, increasing yield through mechanical power, fertilizers, and irrigation to overwhelm these environmental limits. Future yield improvements rely on microscopic precision. Researchers are now attempting to modify rubisco to ignore oxygen or optimize redox pathways to bypass these specific kinetic bottlenecks. Carbon fixation is a constrained thermodynamic engine. Identifying precise limits dictates how we measure and potentially expand the carrying capacity of the planet.